Vesicular transport is one of the three ways of moving molecules around the cell. The other two, as you will remember, are gated transport and transmembrane transport. Vesicular transport uses small balloons of membrane, roughly 40 to 80 nanometers in diameter, which on the football scale is 0.5 to 1 millimeters in diameter, to move proteins, lipids and some other molecules around the cell. Now, before we go into this any further, we need to define two key terms, and those are endocytosis and exocytosis. Cells need to move things in and out of them, and the problem we have is the membrane barrier. We've already discussed some of the solutions to this problem, namely pores and receptors, but a third solution is vesicular transport. So what sort of things may the cell need to move in, or to give its correct name, endocytose? Well, it may need to move in any number of different types of molecule that the cell needs to live, or even relatively large particles. In terms of moving material out, which is called exocytosis, the cell may need to deliver newly synthesized molecules to the exterior, for example proteins, or it may need to deliver newly synthesized proteins or lipids to the membranes. Therefore, exocytosis is defined as moving things out, think exit, as in exocytosis. So that would be transport from the ER to the cell membrane via the Golgi and the endosomal system, and transport from the ER to the lysosomes. Endocytosis, think enter, as in endocytosis, is the transport into the cell from the cell membrane to the lysosomes and the endosomal system. Endocytosis can be further broken down into three types. Phagocytosis, the ingestion of large solid particles into the cell. Pinocytosis, the ingestion of liquid into the cell. And receptor-mediated endocytosis, the controlled import of molecules into the cell using receptors to recognize the molecule. Now, once the material has been endocytosed, there are three possible outcomes. 1. Recycling. The proteins and lipids are retrieved from one compartment and returned to another. 2. Transcytosis. The endocytosed material is transported from one side of the cell to the other. That is, it is taken up on one side of the cell, trafficked across the cell and then released on the other side. And an example of this can be found in cells that line the intestines. They will move nutrients across the cells from the gut and into the body. And 3, degradation. Here, the endocytosed vesicles will fuse with the lysosome, and the enzymes in the lysosome will degrade the material. The lysosomes, as I've said before, are an excellent example of the importance of compartments in cells. The lysosomes have a pH of 5 and contain nucleases, proteases, glycosidases, lipases, phosphatases, sulfatases and phospholipases, all enzymes that break things down. And besides endocytosis material that can get into the lysosomes can also do autophagy, which is when the lysosome engulfs organelles that are no longer needed by the cell. For material that is being exocytosed, there are two main pathways to be considered: the constitutive and the regulated. In the constitutive pathway, things just happen. That is, it is a constant trafficking and it just keeps happening, and once something is on that pathway, it's going to run its course. It will be exocytosed. In the regulated pathway, there is control. That is, the pathway only runs to its conclusion if there is a signal to trigger the pathway. Examples of this are secretory vesicles that release material when triggered, for example, the release of insulin from the beta cells of the pancreas in response to elevated blood glucose levels. Another example of regulated trafficking is the insertion of proteins into the plasma membrane to change the function of the membrane. An example of this is the regulated trafficking of vesicles containing the glucose transporter GLUT4 to the plasma membrane, so more glucose can be taken up by the cell. In this case, the trigger is the activation of the insulin receptor by insulin. Okay, so we've now looked at trafficking of vesicles in the cell. However, we have not addressed two main problems. 1, how are vesicles formed? And 2, how do the vesicles recognise and dock and fuse with their target membrane?